First of all, I have to thank the organizer for giving me the opportunity to give this talk today. And I have to make upfront a disclaimer. I am a pediatric immunologist, and I don't know anything about pulmonary hypertension. But I must say that I learned a lot today. And I also learned that there is still quite a confusion between the role of the adaptive immune system versus the regulatory T cells in the pathogenesis of the disease. And I hope that my talk will give you some inspirational thought of how to go about to investigate the regulatory T cells and the possible dysregulation in the regulatory T cells in pulmonary hypertension. So we know very well that immunomediated diseases are a variety of diseases, and the list is getting bigger every day. And of course, pulmonary hypertension is not in the list because you cannot see it. It was the last one in the bottom with a question mark. So what we do today is to treat these diseases with anti-inflammatory and immunosuppressant. And basically, by doing that, we downregulate the physiological mechanism to counteract inflammation and adaptive immune responses to self-antigens. Indeed, almost all the immunosuppressants we use today block T regulatory cells induction and function. Keep this in mind because maybe we have to get a little bit more sophisticated in that. And indeed, when we think about induction of tolerance, we have to be very well aware that tolerance is not a lack of immune response. It's just a different type of immune response that our immune system is set to put in place when we want to tolerize an antigen, whether it's a self-antigen or a de novo antigen. Think about all the de novo antigen we are daily exposed and we tolerize. Unfortunately, today, despite many, many efforts, there are no drugs out there that really specifically induce tolerance to a specific antigen that we want to downregulate. But we have some indications. So just to refresh your memories, tolerance can be central tolerance in the thymus, and I think that Mark Davis really contributed to show that this central tolerance is indeed an important mechanism, but not the only mechanism. And each of us has in the circulation and in lymphoid organs a lot of autoreactive T cells that are kept under control in normal life. And how are they controlled? Well, there are a number of peripheral tolerance mechanisms that can be put in place. And definitely, regulatory T cells and the suppression mediated by regulatory T cells is an instrumental mechanism of peripheral tolerance. So over the past 20 years, we learned that indeed regulatory T cells play a key role in preventing pathology. And when you have a dysfunction in regulatory T cells, specific for defined antigens, you have type 1 diabetes, IBD, rheumatoid arthritis, etc. So all these diseases are linked to a specific dysfunction in antigen-specific regulatory T cells. On the other hand, we know that regulatory T cells can play a detrimental role in some type of parasitic infection and viral infection. Indeed, we know very well that, for example, if you have hepatitis C and regulatory cells are induced too early, this is contribute to a chronic infection. But definitely, T-regs play a key role in immunological homeostasis. Now, I heard a lot about T-regs today, and I would like to set the stage by saying the CD4 positive T-regs are only one subset of regulatory T cells. There have been NK regulatory cells, CD8, gamma delta, etc. And even within the CD4 positive cells, there are at least 4 or 5 subsets of regulatory T cells. So there are the thymic-derived FOXP3, which are defined by a set of events in the thymus. They are thymic differentiated, and they leave the thymus with a fixed repertoire of TCR receptor antigen specificity. And we think that these are the major players in maintaining tolerance to self. Then you have the peripheral Treg that are induced in the tissue, mostly through TGF-beta production. And these cells are superimposable to the thymic derived in terms of function, but they are very different in terms of markers and also requirements for their function. And then we have the adaptive regulatory T cells, which derive from the naive T cells exactly as the Th1, Th2, and Th17. The difference is the signal that the naive T cells receive. So for example, the Th1 cells, that are the cells that we described many years ago at DNX Research Institute here in Palo Alto, these are cells that are induced when the naive T cells encounter the antigen in a tolerogenic environment where IL-10 is produced. So IL-10 is a master inducer of TR1 cells. And IL-27 is important for the induction of TR1 cells in the mouse, but in the human, it doesn't seem so instrumental. And finally, we have the Th3 Treg that are induced during oral tolerance. So many cell type, 1, 2, 3, 4, at least in the CD4 positive T cell population. So when we think about the FOXP3 positive Tregs, I just want to recapitulate what we know today about the human. So these cells express very high level of FOXP3. But please keep in mind that when you look in the sample of a patient, every single human activated T cells upregulates FOXP3. So FOXP3 is not a specific marker for Treg, especially if you have a lot of inflammation and activation. You have to go much deeper than that. But nevertheless, these cells express high level of FOXP3, and their function is IL-2 dependent. So if you don't have IL-2, they do not suppress. But we still do not know the mechanism of suppression. We know that they require IL-2, they signal through STAT5, but we do not know exactly the signaling between the effector cells and the Treg that leads to downregulation of the effector cells. We know that you require cell-cell contact, but the rest is still mystery. Also, what we still do not know yet is how to induce this is a little bit, that doesn't matter. How to induce these cells in an antigen-specific way. So we have this repertoire of FOXP3 positive T cells, which are regulatory T cells in the peripheral blood, but we do not know about their antigen specificity, and also we cannot manipulate their antigen specificity. But what we know is that FOXP3 is the master regulator, and you need sustained expression of FOXP3 to have a Treg. So if you downregulate FOXP3 expression in the Treg, you lose the function. And the other thing we know, which may be relevant in the context of the disease you are investigating, is that rapamycin is very good in expanding FOXP3 positive Treg in vitro and in vivo. So I just show you this experiment in which we culture the cells in the presence of rapamycin, and you can see that you basically maintain the FOXP3 expression in the cells with rapamycin, but you lose it when you expand the cells without rapamycin. So rapamycin favors the expansion of the FOXP3, and more importantly, Manuela Battaglia, in my group in Milan, showed that rapamycin prevents the skewing of the FOXP3 positive cells towards Th17 in inflammatory environment. So when you have inflammation, you have some plasticity of these cells. And a subpopulation of the FOXP3 can become Th17. Now, if you have rapamycin in the culture, this doesn't happen. And this doesn't happen also in vivo when we give rapamycin to the mice. Importantly, we showed that rapamycin in patients expand the FOXP3 positive cells. This was shown in a type 1 diabetes patients that receive rapamycin prior islet transplantation, and also in patients undergoing bone marrow transplantation. And I just want to remind you that in addition to being an immunomodulant and to favor the expansion of the FOXP3 positive cells, rapamycin has many other effects, including a very strong anti-inflammatory effect, as it was shown by many groups in a medicated stent implant. So rapamycin is definitely a drug which is permissive for Treg expansion and for tolerance induction. So based on what we know about regulatory T cells, of course, we thought that a very simplistic way to cure a patient and to induce tolerance was to shift the balance between the effector cells and the regulatory T cells. So to give more Treg with FOXP3 positive Treg with cell therapy, and really to reestablish a balance between regulation and effector function. And based on this rationale, there have been a number of clinical trials that have been conducted and also completed. And I just want to summarize to you very briefly where we are. So the initial trial, which indeed was also conducted at Stanford by Rob Negrin, was to give freshly isolated CD25 bright Treg to prevent graft-versus-host disease. The trial showed safety and some indication of reduced acute graft-versus-host disease. And then there are 3 other trials with ex vivo expanded CD25 positive cells. This trial used also rapamycin to expand the cells. And in all these trials, again, they showed feasibility, safety, and some level of efficacy, but not dramatic effect. And one of the problems, of course, of all these trials, that the cells were polyclonal, were not antigen-specific. So you don't know if the lack of dramatic effect is because, indeed, you didn't give the right cells. But I still think that we have to distinguish between autoimmunity due to dysregulation of antigen-specific Treg and genetic autoimmune disease in which the dysregulation of the Treg is not antigen-specific, is broad across the repertoire. So from what I heard today, I still don't know if in pulmonary hypertension you think that there is a dysregulation of the Treg, which are specific for a certain antigen, a retrovirus or another antigen, or you have a genetic autoimmune defect with dysregulation in the whole repertoire of the Treg. The situation is very different, also in terms of therapeutic approach. So the prototype of a genetic autoimmune disease is the IPEX syndrome, which stands for immunodysregulation, polyendocrinopathy, and enteropathy X-linked. So in these children, we have a defect in FOXP3. And the classical triad of symptoms, which really bring the patient to death in the first year of life if we don't take care of them, are very severe dermatitis and hyper-IgE due to dysregulation of Th2, very severe diabetes. I'm sorry, there is something wrong with the format here, but I hope you can see it anyway. And very severe enteropathy. And also you have a number of other manifestations in the lungs, recurrent infection, and autoimmunity to target organs. So this disease is due. This is a little bit a problem. I thought that the MAC to MAC should not happen, but that's okay.
I'll take care of it. Go ahead and keep presenting.
I can go on, doesn't matter. So what you can see in these patients is really a dysregulation of the Treg. Sometimes FOXP3 expression is completely eliminated, abrogated. They are comparable to the knockout mice. Sometimes there is residual FOXP3, but what is really the hallmark of the disease is that you have a dysfunction in the Treg. It doesn't matter. I just continue. So, and the mutation can be in different sides of the gene, but there is no genotype-phenotype correlation. What is important to say is that what we learn a lot for this disease, based on the study we did with Rosa Bacchetta, is that pathogenesis is due to different dysregulation. So first of all, I showed you that although the T-reg lineage commitment is preserved, we have a severe impairment in the T-reg function. But in addition to this, what we have is that the naive cells are really skewed towards Th17, and also a proportion of the T-reg which are mutated are skewed towards Th17. We have also an accumulation of autoreactive B cells, and we have massive inflammation with altered cytokine production by the effector cell. So this is a prototype of a genetic disease where a defect in one single gene brings to dysregulation of the old T-reg compartment. I can give you an example of CTLA-4 deficiency, CD25 deficiency, STAT5 deficiency, all the same picture, not the same target tissue. For example, in FOXP3, the target tissue, I told you, is the gut, and is the liver and the pancreas. If you have a CD25 deficiency, the target tissue is the blood and sometimes even the lung. So keep that in mind. The clinical phenotype is different, and we still don't understand completely why. So what we do today for the IPEX patients is basically bone marrow transplantation. But what we are doing, and actually this is planned at Stanford, and this is getting really ugly with these slides, but you know, we are all tired, so we just continue. We are doing gene therapy. So what we plan to do for these patients is to engineer their T cells with a lentiviral vector encoding for the FOXP3 and reinfuse these cells to the patients. So we published that this is visible, results in the correction of the function of these cells. We tested these cells in vitro in the mice. They work, so the next step for us is to go into patients. And we think that by just giving a small subset of these corrected T cells, we can benefit the patient, and if not curing the disease. So I just want to move on and show you some data on the TR1 cells. I didn't hear a single talk today talking about TR1 cells, which was really disappointing. Because I think that the TR1 cells play a key role, especially in diseases which are really where there is a very strong inflammatory component and dysregulation of Th1 and Th2. So the TR1 cells are distinguished from the Th1 and Th2 and Th17 based on their cytokine production profile. They produce very high IL-10 very early on, a lot of TGF-beta. And they are antigen specific. And recently, we identify markers that can identify these cells in the peripheral blood of normal donors and in patients. So please use these markers when you look at your IL-10 producing cells in the samples of the patients. Because this marker will really allow you to identify a population of Treg which is completely different from the FOXP3. Indeed, these cells do not express constitutively FOXP3, but of course upregulate FOXP3 upon activation. IL-10 is the master regulator for these cells. It's the key player in the induction of the TR1 and is the key player in the suppressor function of the TR1. And I just want to remind you that IL-10 is a very strong anti-inflammatory drug. It inhibits macrophage and monocyte and dendritic cells activation, by inhibiting not only a number of co-stimulatory molecule expression, but also by downregulating inflammatory cytokines and chemokines. And also they upregulate a number of natural anti-inflammatory substances that we have in the body, including IL-1 receptor antagonist. Also, what we know is that IL-10 produced by dendritic cells, and specifically by DC10, but also by other dendritic cells or even non-dendritic cells, induce TR1 cells. So again, IL-10 is the major inducer of TR1 cells through this very specific population of dendritic cells. So if you look in the knockout mouse, what you have is a really classical manifestation of the lack of IL-10 and the lack of TR1 cells is exaggerated inflammatory reaction and spontaneous IBD. But of course, we didn't look extensively, for example, at the lung of these mice. What we know is that we can correct this phenotype by giving TR1. And also, if we give TR1 in a model of IBD, we can completely protect the mice from IBD. What is important in these slides is to show that the TR1 cells we give are antigen-specific. And only if we feed the mice with the antigen, in this case was ovalbumin, we really trigger the function of these cells. This is very important because you can think to do an antigen-specific therapy. And the other thing I want to show you is that similarly to the IBD, when you give TR1 cells, you can protect the development of atherosclerosis in the apolipoprotein E knockout mice. Important information, again, from what we want to think in terms of cell therapy. The equivalent of the IL-10 knockout mouse is a human, and this is a rare disease, but again, is a patient's children with genetic autoimmunity due to defect in the IL-10 or the IL-10 receptor. The disease onset is very early. They have severe inflammation, skin, joint, and basically you have to transplant them very quickly, otherwise they die. So the last thing I want to show you is indeed the mechanism of action of these TR1 cells. They need to be activated via the antigen. Once they are activated, they produce IL-10 and TGF-beta. IL-10 and TGF-beta downregulate the inflammatory molecules produced by the dendritic cells, and also the dendritic cells are converted into regulatory T cells. So this is an antigen-specific suppression. But in addition, what they do, they secrete granzyme B and perforin, and they kill myeloid cells, downregulating T cell priming. And one point I want to make, I don't want to say that the TR1 cells are more important or less important than the FOXP3, but you have to keep in mind that there are strong evidence from our group and also from other groups that these two populations have different functions. So what we think is that the FOXP3 are there to induce tolerance. They migrate very quickly in the target organ when you have an insult. The TR1 cells are there to maintain tolerance, and they reside in the spleen in the long-term tolerant mice. So if you wish, you can think that the CD25 FOXP3 are really the firefighters, and they try to switch off the fire and the inflammation as quickly as possible. The TR1 cells are there to patrol and to make sure that the immune system is in balance. And indeed, you can have many papers out there. This is just an example. They show the TR1 cells, high frequency of TR1 cells is associated with tolerance. In transplanted mice, in patients with celiac disease in remission, in patients successfully treated for house dust mite allergy, and also in healthy individuals. So TR1 cells are definitely important in maintaining the homeostasis. And as I told you, we can generate TR1 cells in vitro using different approaches, but more importantly, using DC10, which are the specialized antigen-presenting cells that induce TR1. So DC10 produce very high level of IL-10, express high level of HLA-G, which is involved, of course, in fetal maternal tolerance, and high level of ILT4. And again, we can think to use TR1 cells for clinical trials. And in the next slide, I summarize the trial that have been already completed. This trial was completed in my team back in Europe. We gave TR1 cells to patients receiving haploidentical hematopoietic stem cell transplantation. Another trial was already concluded in Crohn's disease. Both trials showed safety and also some level of efficacy. More importantly, in the trial that we conducted in Milan, we treated 12 patients, and in all the patients, we saw a tolerance signature. And again, I apologize for the slides, but basically what we saw in this patient was a steady and significant increase in the CD49B positive, LAG3 positive TR1 cells. So I'm done. I hope I convinced you that Tregs are important. And I basically think that when you think about an inflammatory and also an autoimmune disease, you may think that both subsets of Treg that I described to you may play a very important role in downregulating both inflammation, especially if these are TR1, because IL-10 is a very potent anti-inflammatory molecule, but also in downregulating the adaptive immune response. And again, I think that it would be important in the context of the disease you are investigating to understand if there is really a general imbalance in the Treg due to a genetic mutation, or this is really an autoimmune disease due to a defect in a population of Treg which is antigen specific. And I thank you for your attention. I would like to thank Rosa, who moved with me to Stanford to investigate the genetic autoimmune disease due to Treg dysfunction, and Silvia, who is still in Milan to conduct the clinical trials. And I thank you for your attention.
So we're actually going to combine the last talk and the panel to consolidate a little bit, so I think maybe we have time for a couple questions.
Yes.
Yes. No, thank you very much. So, do you think that in order for to quell a primed immune response in a vigorous autoimmune process, that you need to potentiate Treg immunotherapy with adjuvant immunosuppression?
Yes, the example is, for example, the trial that Jeff Blueston at UCSF is planning to do as a follow-up of the initial trial in the type 1 diabetes, where with the FOXP3 Treg, they plan to give rapamycin in vivo to the patients. For the TR1 cells, certainly one possibility is, to give in conjunction with the TR1 recombinant IL-10. Now, as you know, recombinant IL-10 failed in the clinic because per se is not an immunosuppressant, it's an immunomodulant. So the company that developed IL-10 positioned IL-10 as an immunosuppressant, whereas, again, doesn't have any anti-proliferative effect at all. It just has an immunomodulatory effect.
Great, and just a quick follow-up. You were disappointed that we weren't delineating TR1s from Tregs. We don't know if the genetic defect, the main one is BMPR2 mutations and heterozygosity in patients. We don't know if that's contributing, but there are some clues that actually that BMPR2 heterozygosity may impact T cell development. If you BMP4 knockout mice, which is part of the signaling cascade, BMP4 signaling is required for normal T cell education in the thymus, and it's also BMP signaling is also important for B cell activation. So we don't really know if this is environmental or genetic at this point. So
I understand that. I learned that. Yes.
Are there any markers that are expressed in conjunction with the transient elevation of FOXP3? And the second question, when you look across a broad spectrum of autoimmune diseases, in which of them do you see impaired TR1 cells?
First question, thank you for asking that. Actually, I should have said that. Basically, the only assay that really prove, that you are looking at thymic-derived FOXP3 positive cells is to look at the TSDR tandem repeat of the gene. This is quite difficult. I mean, we do it, but you need a specialized lab. So what it may be much easier is to take these cells and to see if they're functional in vitro. Because, if these are activated T cells, for sure they do not suppress. effector cells. Whereas if they are FOXP3 positive cells that are really bona fide Treg, they suppress in vitro. But I just think that all the markers, for example, you can look at CTLA4, you can look at PD-1, you can look at GITR. All these markers are also upregulated in activated T cells. So at the end of the day, you're looking at a difference in MF5 and in the level of expression, which can be a little bit tricky. So I would suggest do a functional assay, prove that these cells are suppressor in vitro, and that's good enough. The second question, actually, the patients, the autoimmune disease where we saw, not only autoimmune, but also inflammatory disease, where we saw a dysregulation in TR1 go from, organ rejection, graft-versus-host disease, also type 1 diabetes, celiac disease, as I mentioned, etc. But the genetic disease with the impairment of the IL-10 or the IL-10 receptor is really the disease that shows that if you lack these cells, you have arthritis, you have IBD.